Welcome to the microscopic battlefield. In this opening scene, two very different cells stand side by side. One is large, complex, and confident, a eukaryotic cell that proudly declares it has a nucleus and mitochondria. Next to it stands a much smaller bacterium, quiet and simple, admitting that it does not have a nucleus. Despite its simplicity, this tiny cell is capable of causing massive disease, reminding us that size does not determine danger in microbiology. Life is divided into two fundamental cellular worlds, prokaryotes and eukaryotes. Prokaryotes, which include bacteria, are simple and small cells. They lack a true nucleus and do not possess membrane-bound organelles. Their DNA lies freely in the cytoplasm, and their streamlined structure allows for rapid growth and division. In contrast, eukaryotic cells are larger and more complex. They contain a true nucleus, mitochondria, endoplasmic reticulum, lysosomes, and other organelles that compartmentalize cellular functions. This difference is not only structural but also clinically significant. When we focus on the control center of the cell, the contrast becomes even clearer. In eukaryotic cells, DNA is enclosed within a double membrane nucleus and organized into chromosomes. In bacterial cells, there is no nuclear membrane. Instead, genetic material exists as a single circular double-stranded DNA molecule located in a region called the nucleoid. The absence of a nucleus and histones allows bacteria to replicate and adapt very rapidly, which explains how bacterial infections can spread so quickly in the human body. A direct comparison highlights these differences further. Prokaryotes lack a nucleus, contain circular DNA, and do not have membrane-bound organelles. They are small in size, typically measuring between 0.5 and 5 micrometers, and possess 70 ribosomes. Eukaryotic cells, on the other hand, have a true nucleus, linear DNA, multiple organelles, a larger size ranging from 10 to 100 micrometers, and 80 ribosomes. These distinctions are essential for understanding both cell biology and clinical microbiology. These structural differences are exactly why antibiotics can selectively target bacteria without harming human cells. Many antimicrobial drugs are designed to interfere with bacterial features that do not exist in eukaryotic cells, such as the bacterial cell wall, 70 ribosomes, and specific enzymes involved in bacterial DNA replication. This selective toxicity is the foundation of effective antimicrobial therapy. Bacteria come in different shapes, and these shapes are one of the first clues we use in identification. Some bacteria are round and appear as cocci, which may arrange in pairs, chains, or clusters. Others are elongated rods known as bacilli. Some bacteria twist into spiral forms called spirilla, while others curve gently into a comma shape, known as vibrios. These shapes are not random. They influence motility, attachment, and pathogenic behavior, making morphology a key starting point in clinical microbiology. In the laboratory, bacteria are first differentiated using the Gram stain, a fundamental diagnostic tool. After applying crystal violet and a counterstain such as safranin, bacteria appear either purple or pink under the microscope. Those that retain crystal violet are classified as Gram-positive, while those that lose it and take up safranin are Gram-negative. This distinction immediately narrows down possible pathogens and guides early antibiotic choices. The reason behind the Gram stain result lies in the structure of the bacterial cell wall. Gram-positive bacteria have a thick peptidoglycan layer that traps crystal violet, causing them to appear purple. Gram-negative bacteria have a thin peptidoglycan layer and an additional outer membrane, which allows the crystal violet to be washed away, leaving them pink after counterstaining. Thus, Gram reaction reflects architecture, not color preference. We'll now discuss in detail various bacterial cell structures. The Gram-positive cell wall is dominated by a thick peptidoglycan layer, often measuring 20 to 80 nanometers. Peptidoglycan is a strong mesh-like polymer composed of alternating chains of N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM, cross-linked by peptide bridges. Embedded within this wall are teichoic acids and lipoteichoic acids, which contribute to structural stability and play a role in immune activation. This unique cell wall structure provides rigidity while allowing controlled growth and division. Because peptidoglycan is absent in human cells, it is an ideal target for beta-lactam antibiotics such as penicillins and cephalosporins. Gram-negative bacteria possess a more complex cell envelope. Beneath the outer membrane lies a thin peptidoglycan layer located within the periplasmic space. The outer membrane contains lipopolysaccharide, or LPS, which functions as an endotoxin and is a major trigger of fever, inflammation, and septic shock. Porin proteins within this membrane regulate molecule entry, contributing to antibiotic resistance and making gram-negative infections particularly challenging to treat. Beneath the cell wall lies the cytoplasmic membrane, a phospholipid bilayer that plays a vital role in bacterial survival. It controls selective permeability, allowing nutrients to enter and waste products to exit. Unlike eukaryotic cells, bacteria generate ATP at this membrane rather than in mitochondria. It is also involved in secretion, transport systems, and cell division. Clinically, this membrane is the target of polymyxin antibiotics, which disrupt its integrity. Mesosomes are inward folds of the bacterial cytoplasmic membrane that increase surface area. They are traditionally described as sites for DNA attachment during cell division and for the concentration of respiratory enzymes. Although their existence as true structures has been debated, mesosomes remain a classic concept in basic microbiology, enabling us to understand how bacteria compensate for the absence of mitochondria. At the center of the bacterial cell lies the nucleoid, which contains a single circular, double-stranded DNA molecule. This DNA is supercoiled and not surrounded by a nuclear membrane, allowing rapid replication and transcription. The enzyme DNA gyrase maintains this supercoiling, and it is precisely here that fluoroquinolone antibiotics act. This organization explains the speed at which bacteria grow, mutate, and adapt. Plasmids can also be found in many bacteria. A plasmid is a small, circular, double-stranded DNA molecule, separate from the main chromosomal DNA. It is located in the cytoplasm and has the ability to replicate independently of the bacterial chromosome. Plasmids are not essential for basic bacterial survival, but they provide important adaptive advantages, especially in hostile environments. One of the most clinically important features of plasmids is their ability to spread between bacteria through horizontal gene transfer, particularly by conjugation. Inside the bacterial cell, like eukaryotic cells, protein synthesis takes place on ribosomes, which function like miniature factories. Bacterial ribosomes are composed of two subunits, the 30S and the 50S, which come together to form the 70S ribosome. Amino acids are assembled into protein chains that are essential for bacterial growth, metabolism, and virulence. These ribosomes are structurally different from human 80S ribosomes, a difference that is clinically exploited by many antibiotics such as aminoglycosides, tetracyclines, and macrolides. Bacteria often store essential materials inside the cytoplasm in structures known as inclusion bodies. These storage granules allow the cell to survive periods of nutrient deprivation. Glycogen granules store energy, lipid inclusions act as fat reserves, and velutin granules serve as phosphate storage. Velutin granules are particularly important in microbiology because they are classically associated with Corynebacterium diphtheriae and can be demonstrated using special stains, making them a useful diagnostic clue. Some bacteria have extra protective coat, the capsule. The bacterial capsule is an outermost protective layer that surrounds the cell wall in certain bacteria and acts as an invisibility cloak against the host immune system. This capsule is usually composed of polysaccharides and makes the bacterial surface slippery, preventing effective recognition and engulfment by phagocytic cells such as neutrophils and macrophages. As a result, encapsulated bacteria are significantly more virulent than their non-encapsulated counterparts. In addition to its antiphagocytic role, the capsule enhances adherence to host tissues, helping bacteria colonize and establish infection. Because it interferes with immune clearance, the presence of a capsule is often associated with severe invasive diseases, including pneumonia, meningitis, and septicemia. Clinically, the capsule corresponds to the K antigen, which is used in bacterial identification and serotyping. Motile bacteria have flagella. Flagella are long, whip-like structures that provide motility, allowing bacteria to move toward favorable environments and away from harmful conditions. Bacteria may possess different flagellar arrangements, including monotrichous, lophotrichous, amphitrichous, or paratrichous patterns. Beyond movement, flagella play a role in pathogenesis by aiding bacterial spread within the host. Clinically, the flagellar protein flagellin is an important antigen known as the H antigen. Pili are short hair-like surface structures with two major functions. Adhesive pili, also called fimbriae, allow bacteria to attach firmly to host tissues, a critical step in colonization and infection. Other pili form the sex pilus, which creates a bridge between bacteria during conjugation, enabling horizontal gene transfer. This process is a major mechanism for the spread of antibiotic resistance genes among bacterial populations. Some bacteria can enter an extreme survival state by forming endospores. This ability is limited to certain genera, most notably Bacillus and Clostridium. Endospores are highly resistant to heat, chemicals, radiation, and desiccation, allowing bacteria to persist in harsh environments for long periods. Importantly, endospores are not reproductive structures but survival mechanisms, and they are destroyed reliably only by autoclaving. Alright, let's recap. Each bacterial structure serves a specific function that directly influences clinical behavior. The cell wall provides shape and protection and is a major target for antibiotics. The plasma membrane regulates transport and energy production. The nucleoid contains genetic material, including virulence genes. Ribosomes synthesize proteins essential for survival. Capsules inhibit phagocytosis, flagella aid motility, pili enable attachment, and endospores ensure survival. Understanding these relationships is key to mastering microbiology. Ultimately, understanding bacterial structure guides clinical decision-making. Antibiotics are chosen based on whether they target the cell wall, ribosomes, DNA replication machinery, or membranes. By recognizing which structures are present and functional in a given bacterium, clinicians can select the most effective treatment and improve patient outcomes. In microbiology, structure is not just anatomy, it is the roadmap to therapy.